Thank you, Mr. Rector. Dear candidate, let me start with complimenting you with the completion of this wonderful thesis. You performed an enormous amount of work to actually improve the outcome of next generation sequencing in diagnostics. And you've also learned us what the challenges and are still there still are. And I think it's also a wonderful start for what your ambitions is to use these techniques, hopefully also in your homeland, Tanzania. So let me start with first questions in terms of challenges. So in your introduction and in your discussion, you mentioned a couple of times, of course, that there are advantages of next generation technologies, but also limitations, first in diagnostic yield, throughput, and costs. But then you also mentioned that the choice of next generation sequencing platforms for routine testing is also important when you think about a contribution to clinical utility. And actually, you don't really elaborate much on that. So I would like to ask you to do that now. First of all, what do you mean with clinical utility? What is your definition? And then what is actually then the contribution and how would you elaborate on the use of which platform you use for clinical utility?
Highly learned opponent, thank you for your kind words and for the very interesting and good question. To start off with, yes, indeed, we have multiple platforms that use next generation sequencing to detect genetic variants that have a causality for a particular disease. It is very important first to understand from a literature point of view what are the different types of variations that are present within the genome that cause that particular disease, and then subsequently how much of it do we actually are able to capture using those particular techniques. And each of those techniques, of course, has their own pros and cons in terms of what it can capture and what it has limitation in terms of not able to capture. And due to that, it is important to understand the genetic variation. For example, if you have large structural variations, what are some of those techniques that would be able to capture the large structural variations, which you would not be able to, for example, with short read sequencing?
Sure, but let me just ask you if the way you seem to go now is about diagnostic yield.
Yes.
And that's, in my view, something different than clinical utility.
Yes.
You agree?
So the definition of clinical utility, in fact, has a lot to do with the downstream costing as well, as far as the value to the patient himself, because diagnostic yield is one thing, and then the clinical utility is actually following up on the patient in terms of how the therapy will affect or how that particular diagnosis will help the patient in terms of the therapy for that particular patient for that disease. And that is something
Does it? If you have, let's say, if you find a really pathogenic variant in a cardiomyopathy gene.
Yes.
Does this change the management of the patient?
It helps with the screening of the family to start with. That is very, very important because if at all we are able to screen for a specific mutation within the family, then we know if the patient will require lifestyle changes or already in starting of certain medications beforehand to help to sort of delay the process of getting the particular disease. And that already helps the patient in terms of the clinical utility.
But if you consider the fact that you also find so many VUSs, especially in, genes related to cardiac diseases, is that really always beneficial, or can we also sometimes harm the patient with these kinds of, answers to their questions?
Depends on what we find. So for instance, if you have a variant that we don't know the clinical significance, which we classify as a FUS, that can increase anxiety levels of the patient because now they really don't know what caused that particular disease. But in the case where we are able to find a genetic variant, then in most cases for the studies that have been done, it has shown to help the patient, first of all, understand what caused the disease. That in itself already relaxes them in terms of their anxiety, nervousness. And then hopefully, when enough knowledge is available, they are able to get the right kinds of therapy or treatment, or even lifestyle modifications to cater for that.
But if we then should go, you say it's also important for family testing. I agree there, so cascade testing. But I remember a few years ago we had a large discussion with the governmental department that looks at costs of testing and whether it not should be insured.
Yeah.
And so they said, well, given the fact that not all of these variants are actually, fully penetrant, you might also, by doing this whole cascade testing, and let's say you find the variant in a family member who's, still doesn't have anything, you put them into follow-up for something that may never occur. So again, they asked us the question whether this is, whether this is really beneficial to do this cascade testing. What would be your answer to this department?
I think we need to see the bigger picture here, that it's sometimes not just about one particular family or one particular patient. We have to see it as a global agenda or knowledge contribution to the genetics field, because it may not necessarily directly have an impact on a patient at that particular point of time, but through that particular experiment, that knowledge might contribute to diagnosis of other patients that might have similar mutations that, that would be causative. So I think from a bigger picture, that would be quite relevant.
Yeah, I agree there with you, but I'm afraid the department will not think like this, but I fully agree. I'm happy with your answers and give the word back to the director. Thank you.
Thank you very much. The discussion will be continued by Professor Ramsey.
Thank you very much, and good morning, learned colleagues. And Mohamed, how lovely to see you there. And I wish to thank you for the excellent thesis that you've given me and everybody else to read. Really seeing your progression over the years of your PhD has been very gratifying, and seeing the end product is excellent. So I'd like to ask you two things. You and I both come from Africa. You from Tanzania. I'm from South Africa, at the moment sitting in Abuja in Nigeria. But I want to ask you, next generation sequencing is quite a sophisticated technology to implement. And what do you think the critical factors are for trying to implement a clinical genetic service in a low income setting or low resource setting like you have in Tanzania?
Highly learned opponent, thank you very much for that very interesting question, very relevant. I think we should draw certain inspirations from countries that have already done so. And in particular, for example, in Africa, we have seen Egypt and South Africa with quite a good genetic service available in the country. And other countries, of course, such as Rwanda and other places where these facilities are now becoming more and more available. So what we need to do there is to draw inspiration from those countries. And my focus, or my idea, would be to focus on two things. One would be to train our scientific students, so basically in the lab, but also bioinformatics. And two is to focus on implementation of or investment in the bioinformatics or data analysis area, because the NGS as a service is always going to change. The technologies are rapidly evolving, and we don't necessarily need to have the sophisticated state-of-the-art technologies in the country, but as long as we are able to outsource them in a way that we are able to get our own data for analysis, I think that has more value in terms of being able to diagnose and look for certain mutations that are peculiar to the African population in the African continent. That would have, in my view, would have more value. And I think a very good example is how Sudan has done it by really outsourcing the NGS itself, but the bioinformatics has really been established in the country. That would be my approach.
So I think, you're right that the analysis of the data is really critical. But what is the infrastructure that you would need? Because, of course, as bioinformaticists, geneticists, we don't work in isolation. So what do you think are the other elements that would be necessary to make this a success?
So in terms of really starting up such facility, first of all, we would have to have infrastructural capacity building, and that really has to ensure that we have a biobank. We have we've taken care of the power outages that are quite common in African countries. We have significant investments in specific areas that are in particularly required for lab experiments and those kinds of tools that would allow us to sort of investigate these kinds of genetic disorders. But at the same time, I think we should not leave out the training component out of this discussion, because you'd have to really invest in training people, but at the same time also the infrastructure, as I think both of them go hand in hand to really establish these facilities.
Yes, and I think the broader infrastructure in terms of the the health system is really important, because you need clinicians who will, first of all, refer the patients, and who can then sort of give you clues about where to look or what to look for. So I think, it's that whole infrastructure that we really need to focus on. But I wanted to take you, Mohamed, you've trained now in the Netherlands, and most of the data that you've used has been on people of European origin. What do you think are the things that will be different when you start looking at patients from Africa? What are the things you will need to think about when trying to find pathogenic variants?
Well, for a start, I think most of us will agree that the African population data is severely underrepresented in various genomic databases. So to really be able to provide a diagnosis in the clinic for such patients, we need to first nationally, locally as well, and within African continent, collaborate with institutions like H3 Africa and various other studies that have taken African cohorts to understand the different types of genetic variants that are present in the genome, because it's no secret that African populations have the greatest number of genetic variants in their DNA. And so that would be the first step towards really being able to classify, because we are then able to compare the frequencies of those genetic variants between patients and then the healthy population. And that would give us an idea of, whether that particular variant is in fact causative of the disease. But at the same time, what would also be interesting, in my opinion, would be that if we were to take a specific disease from which, in my case, as you asked, for example, for my cohort, the cardiomyopathy cohort in the Netherlands, we looked at various different subtypes of cardiomyopathy. The interesting aspect is how many of these targeted sequencing panels that we've used for diagnosis would actually be beneficial to provide diagnosis of the patients, for example, in Tanzania, whether the same genes pop up when we do the whole targeted sequencing, and then if yes, what would be sort of the difference in the type of variation or even the location of the variation within the locus or the specific gene that is already known to be implicated to cause a specific cardiomyopathy subtype. And the same thing would also imply, for example, for diseases like ataxia and other types of genetic disorders, where we already know from literature what's important, but we don't know how different the continental genotype will be from the rest of the world. And that would be really interesting to see.
I think you're quite right that we need to generate more data on patients from Africa, because only then can we understand, locus heterogeneity as well as allelic heterogeneity in disease causality. So an awful lot of work that needs to be done in Africa on African participants. But when you go back to Tanzania, how would you think about prioritizing? As you say, you've worked on genes for cardiomyopathy, but what would you focus on in the first instance and build up a base of research data from? Which disease or group of diseases?
Maybe can you give a short answer to this question?
Yeah, I think my approach would be to go with the low-hanging fruits. So really looking at, for example, diseases that would not require extensive next-generation sequencing data, maybe look at more cytogenetic data, because that's something that's directly implementable. And I would look at sickle cell disease as one of the model monogenic diseases to see how certain types of diseases can be investigated utilizing what the continent has done as far as sickle cell is concerned.
Thank you very much, Professor Ramsey. It's time now.
Thank you.
Thank you very much for your questioning and that you were able to join us. And please stay with us. We'll continue our discussion with Professor Schwering from the UMCG.
Thank you. Dear candidate, first also I would like to compliment you with this very nice thesis, and I also would like to congratulate your team of supervisors with this success. I enjoyed reading your thesis very much with very interesting data, and at least it gave me a lot of new insights, especially on the classification of these buses. Since this is a growing group of variants, because we are now performing more wider and broader next-generation sequencing, we see more and more, as you describe in your thesis. Now, I'm a clinical scientist in molecular pathology, as you know, and in fact, my daily work is interpretation and classification of variants. And since we are also doing a lot of broader sequencing, we are also, personally, I'm also struggling with how to deal with this. So my first question, I would like to take you to your discussion where you state on page 189 that these fuses poses difficulties for the lab, for patients, for healthcare professionals. In fact, regarding the communication of these, in fact, unclear findings, how do these, especially the healthcare professionals, deal with that? So in the pathology field, half of the laboratories do not report fuses in the pathology reports, and half of the laboratory do. We are the labs that are not doing it. So how do you do that in the genetic area? Do you report these fuses?
Highly learned opponent, thank you for the kind words and for the very important question. I think there is a little bit of a difference in terms of how we classify variants that are somatic versus those that are germline. And to what I have studied and what I know of is that patients these days prefer to know whether or not they have a fuss. Yes, you can have a debate as opposed in terms of like whether you want to communicate that to a particular patient or not. But I think empowering patients, this is my personal view, empowering patients with information, even though we know they are not yet clinically actionable, is the way forward. Because many a times, for example, as you know, one of the criteria for pathogenicity determination is the family segregation data. And one really good way to get evidence of such segregation data is to, in fact, empower the patient to provide a link to different cousins also. And so that would really help us to get additional evidence by the classification.
You say us, so I understand that. You say also the patient should know.
Yes.
But still, I'm still a bit concerned because your whole thesis is on how to interpret a VUS and whether you can reclassify it. And just mentioning all these data that we do not know what they really mean to a patient. I mean, we in our practice, we sometimes see BRCA mutations in tumors, and then we call it, we classify it as a VUS. And I think if I would put that in the reports, the clinician might interpret, or the patient might interpret that information a little bit different from what the real meaning is, since it's a VUS. So are you not afraid that you're really concerning patients with this kind of information?
Well, not really, because I think we need to see the bigger picture here. I think, as I mentioned, genetics is a field that not only looks at the information from molecular geneticists or clinicians, but I think it is an active involvement of the patients as well. And so maybe we can look at it from disease-specific model on how best to communicate it to the patient. But in lots of cases, my belief is that the patient should be made aware and not, the information should not be hidden, particularly what I have seen in Tanzania, what we have experienced in Tanzania. Patients tend to get only if the result only tells if it's a likely pathogenic or a pathogenic variant. But behind the scenes, we know that there are fusses, and they would like to know that because that gives them that hope that there's still something that can be done.
I understand your point. I mean, that we know, I totally agree.
Yeah.
For the patients, I just would like to give you the message to really rethink that. On the other hand, in your thesis, you described a lot of fusses and do a lot of analysis to get them reclassified. What I learned from this thesis is that if we report a fuss, in fact, this is a moment that we describe this variant in the patient. And in fact, we know that in time, by technology, by other algorithms, etc., that fuss that we classify as fuss today might be reclassified. You show a lot of ways of reclassifying. In fact, should we start doing reclassifying all of fusses for the patients? And how often should we do that? And when should we do that?
Yeah, in fact, that is the broad message of the thesis. In fact, that is exactly what I'm saying, that periodically, maybe every 2 years, as we expect, because see, the answer to really be able to reclassify some of these fusses is the fact that we'll have more data. And that's also why it is so important that underrepresented populations such as the African continent, as well as colored populations, are sequenced as well. So that information gives us the extra knowledge. Because if you look at the statistics now, even the diagnostic yield, we have a large chunk of variants that we don't know what to do with, which is simply unclassified. And so it is important to have those populations so that these additional information is available, and more tools like what we have shown with the cardiomyopathy, for example, the statistical validation of certain methodologies, the constraint metrics, functional evaluation, will definitely contribute towards some of the reclassification.
So if I may, coming back, you say 2 years, that would be a point to reclassify, yes. But the technology is moving so quickly.
Correct.
So is 2 years not too long for in the genetic area?
Well, the technology is moving quickly, yes, but till it gets to the diagnostic application, that takes a little bit of time. So 2 years, I think, is safe.
Okay. Do we have time for another question?
Short question.
No, I don't have short questions, I think. Thank you very much. Thank you very much for the answers. I go back to you.
Thank you very much. Yeah, it's always a question whether you want to have a short question or not. Professor Ploos van Amstel will continue the discussion. He's from the UMCG Utrecht. The floor is yours.
Waarde promovendus, dear candidate, first I would like to congratulate you, of course, with your beautiful work and well-written thesis. Also, my special congratulations to your promoter and both co-promoters and the team. I very much enjoyed reading your carefully constructed dissertation. Identifying variants and interpreting or classifying variants ultimately as benign or pathogenic, that is a continuously hot topic. And you did it both by data analysis and also experimentally. And also I would like to say that I appreciated that you also paid special attention to the social context of genome research and diagnostics, and to the importance of having all population groups and continents share in this equally, genetic equity and fair data. But also I have questions, and I would like to go into more detail on chapter 4 and on page 88. There you describe a variant in the DES gene at position 79, G2A. A variant we also encountered in Utrecht. And on page 112, 112, you show that the results of the minigene experiments, and that seem to be normal, or not abnormal, not deviating from the normal pattern. And also by RT-PCR experiments, you didn't find any disruption. But still you conclude that the conclusion was not decisive, and because of lack of evidence. When is it conclusive, your experiment?
Highly learned opponent, thank you very much for the kind words and for your interesting question. The DES variant that we found in this particular patient, of course, what we did through the protocol that we designed. So this was basically looking at Alamut as a specific tool, and then the algorithms that are present within that, and how to navigate some of those algorithms with certain thresholds. to fine-tune and sort of filter the important strong candidates and the weak ones. And through that, the process, of course, we have to understand the bigger picture here is that VUS classification or variant classification, so to speak, not only takes into account, for example, just the functional data or the in silico prediction, but it also looks at the penetrance, the conservation evolutionary, and of course the frequencies and various other things. In this particular case, we first wanted to use the decision tree to narrow down this particular candidate. That was interesting. And again, even from the mini-gene analysis, we were not able to successfully clone this because of the large flanking regions. And that's also a limitation of a mini-gene sort of platform to really be able to get the construct right into the plasmid and subsequently to transfect it. At the same time, we were thinking if we were able to get the result using the mini-gene assay and then get a similar result from the RT-PCR on the blood sample, that would have additional evidence, not only in silico, but also functionally, to then get more evidence towards pathogenicity. So in the end, it would not necessarily, should the RT-PCR or mini-gene still work, it would not have necessarily been a classified pathogen or likely pathogen variant unless we tick the other boxes within the ACMG guidelines that would be important for the classification.
So, but I ask, when do you decide to stop? To say it has no influence on RNA or the protein expression. You still have some experimental data that underline that it has no effect, or not?
No, not from RT-PCR. So it was, yeah.
And in Utrecht, we also found in the same family a disruptive variant in another gene. Would that help to classify your variant in this case?
Well, it depends, again, if that particular gene is, number one, implicated for that particular cardiomyopathy subtype, and also if that variant is present in a specific functional domain that already is indicative that those kinds of variations in that particular locus or specific region of the gene is an important area for detecting such kind of variants. In that case, it would be interesting to compare.
Okay. And I have another question about the mini-gene experiments. The outcome, as you have nicely illustrated in the different figures, seems to be not very predictable. You can see all kinds of effects for a single variant. And the agarose gels show those different fragments. Can it be that there is some bias in the selection of choice of accents that you will include in your mini-gene experiments?
Well, there are certain limitations also with respect to the mini-gene, and yes, certainly one of them is not, I would not frame it as bias, but I would say what is available at the time. And yeah, that would be some sort of a trade-off, because as you know, these kind of splicing variations are, if I can use the word, tough cookies to crack when it comes to genetic variations. And they are not only tissue specific, but also they have different transcripts that have different expression levels. So yeah, it's really about what is available at the time, what can we test, and what do we get out of that. That would determine whether or not a particular variant is classified or not.
And how about the segregation analysis in the families? Will that be of help?
Certainly. Family segregation is, in fact, very, very important. But the challenge, again, is that a lot of patients' information is either not available, or the family members are unfortunately have passed away, or there are no ways to get back that data from family segregation. So one way to, in fact, mitigate such an issue is, again, like I was discussing earlier, that we empower the patient themselves. And for example, initiatives like the Gene Matcher or even the Find My Variant initiative, where you really give the patient the platform to record their own family tree, pedigree, and information about such kinds of information that would then enable molecular geneticists to classify certain variants.
Mhm. Is there I don't know, do I have still some time for a question?
For a short question, yes.
Yeah, is there a moment in the diagnostic process that we may conclude there is no underlying genetic cause? It's a short question, maybe a long answer.
It's a very...
Maybe you can try also to do a short answer.
In a nutshell, yes.
Thank you.
That's a very good short answer. Thank you very much for that. Okay. The discussion will be continued by Professor Hamel of the Radboud UMC.
Thank you, Mr. Rector. Dear candidate, of course, I would like to start by congratulate you for the nice work you have done. It's about diagnostic use of NGS and in particular about problems related to variant classification, in particularly thesis. Ongera sana. But also I would like to convey my congratulations to your supervisor and your two co-supervisors. Briefly, you mentioned in the general discussion and your propositions the importance of equity, genetic equity and data sharing, and very rightly so. But you're also dealing in your professional life with the so-called rare diseases, which mostly are monogenic disorders, actually. You and others wrote a national call for action. You are the lead investigator of a study to determine the number and type of rare diseases in Tanzania. And in 2021, you presented in the UK as an invited speaker a lecture, a paper entitled The Clinical Genetics Services in Tanzania. And I would like to start my discussions with that topic. Can you briefly summarize what you told those people in the UK?
Sure. Well, that was a 45-minute lecture, but I will try my best to...
So with the emphasis on briefly.
Yes, yes. Well, so highly learned opponent, thank you for those kind words and for your question. The clinical genetic services in Tanzania as of now are not centralized and not formally done. But we have, that is not to say we don't have any clinical services. In the current aspect, we have various UMCs, for example, the Muhimbili University, the KCMC, where you are also a visiting professor, that come across certain patients that are, of course, exhibiting very rare forms of genetic diseases. And it becomes very important not only to give the genotype to that particular phenotype of the patient, but also to sort of provide that evidence to the scientific community. And so with rare diseases, what we want to really establish in Tanzania is, first of all, surveillance, because from the epidemiological point of view, we still don't know what forms of rare disease or what types of rare disease, because that rare diseases is a very broad term with 7,000 different diseases and over 300 million people worldwide. And so we want to know of the rare diseases, if I can say, the more common rare and the rare diseases that are present in Tanzania. And through that, we then want to prioritize the efforts that are being taken by the government, for example, to provide access to some of the orphan drugs that are needed for treatment of such patients, but also to help train clinicians on how to identify them, number one, but also what kind of infrastructure requirements are necessary within the clinics or the hospital to receive such patients, because that in itself is already an issue in low-income countries. And that was what I told them.
Okay. Very good. What sort of obstacles do you see at the moment in order to reach where you want to get in due course?
It's quite multifactorial, but I think in the few years quite doable. Number one is the fact that a lot of the patients themselves don't know that what a genetic condition actually means. And we still have certain cultural stigma related beliefs that prohibit certain parents to come out with these children and say that they have a rare condition that's in fact caused by a genetic variant. So that can be tackled by education to the mass education to the public. Number two, I think willingness from the government to invest in this particular area is something that I think for Tanzania we're doing quite well, because the government is now really proactive. But a lot of countries in Africa really need to do that and establish national strategies on how to really improve the molecular genetics aspect of diagnosis. But also, like I said, infrastructural development and the capacity building of the workforce. That also needs to go hand in hand, because end of the day, if the clinicians are not able to identify those patients, and if the presence of genetic tests is not existent, then there's not little we can do for those patients.
Don't you think so? Don't you think that at this moment, the number of clinicians in the country which will be able to diagnose rare diseases is far too limited, and that is probably a main obstacle to do an epidemiological study to rare diseases at this moment?
Yes and no. Yes, because indeed that's correct, that the number of clinicians are way more fewer than what is required if we look at the ratio of the number of patients each physician needs to account for. That's true. But no, because we have already quite some areas which are rather small, that we can start off as pilot studies, because rather than waiting till we have the right number of clinicians, we can look at smaller spaces. So in Tanzania, for example, we have the island of Zanzibar, which is very, very interesting to look at genetic conditions, more particularly because 98% of the population in Zanzibar is Muslim, and lots of intermarriages. So that would be a really interesting area to look for such surveillance patterns, just so that we have an idea on how to do it, and then we can move to the mainland to see how we can implement it at a nationwide.
So in fact, it all comes down to increase awareness on different levels. Is it public, the general public, the medical profession, and the government?
Yes.
And what role of the Tanzanian Human Genetics Organization do you see there?
So the Tanzania Human Genetics Organization really, I have to thank Professor Michel here, stemmed out from the Africa Society of Human Genetics, which is sort of the mother society of the continent as far as creating awareness of genetics in the continent is concerned. And we operate in sort of four main areas. One is really looking at research. Two is diagnostics. So we want to implement some of the research and training in daily practice. We have the training component, and we have the patient advocacy. These are the four main pillars of THGO. And within that, what we see as ourselves, or rather THGO, is the vision is really centralize the coordination. Because one of the key bottlenecks, if I can say, in countries in Africa, is that you can be in Dar es Salaam, in the University of Muhimbili, and you can be in Moshi at KCMC, but if the two are not data sharing or communicating, then there is a severe lack of understanding of the prevalence of certain types of diseases. So I think the coordination or the centralizing such genetic diseases or the cohorts would be very instrumental if THGO is able to do that. And that is a bottleneck I see.
Asante sana.
The discussion will be continued by Dr. Jongbloed of the UMCG.
Thank you very much, Mr. Rector. Achte promovendus, dear candidate, dear Mohamed. It's great to sit here, to see this nice work in front of me, and I want really also to congratulate you with finishing this thesis. It's a great job that you did. What you focus on is something that we also focus on in daily practice. So what challenges that we have in the outcomes of the genetic diagnostics of patients, and then of course in particular the fuses. And you had, well, you follow different approaches to tackle that, and I think you did that in a very nice way, and also presented in a very nice way, and also not mentioned yet, also in a very collaborative way. And I think a lot of people around here in the room did collaborate with you to come to this nice result. So, of course, there's still a lot of questions to ask, and I want to continue actually on the questioning that was done by Professor Ploos van Amstel concerning the splice variants. First of all, and it was already touched upon, you could argue this mini gene system, it's a not non-biological, not the biological environment, and the genetic environment is also not as it is in the patient itself. So you could argue, well, you get results, but is that related to what's happening in the patient? So how would you defend yourself on that?
Learned opponent, thank you very much for the kind words and for the question. If I understand you well, and correct me if I don't, that what is in fact, the value of the mini gene as far as the classification of splicing is concerned, and where we see the field moving in the future. So yes, at the moment, mini gene is a very important tool. I would argue that for various number of variants that do not fall under the limitations of mini gene are still quite useful to study for the understanding of how the variant actually affects the biological process. But I think moving forward, because of these limitations, more and more labs will probably adopt a more RNA sequencing approach, particularly to look at the expression changes per tissue, probably, with a follow-up of functional testing using mass spectrometry to look at really the protein change. I think also more needs, as we've shown in the SCA spinocerebellar ataxia paper, that protein modeling is another thing that can be added to how we would like to address variant interpretation, but particularly for the splicing variation, probably also high throughput methodologies such as the VecSeq or the MAPSI tools, which are really looking at large number of variations. So
now I'm going to interrupt a moment. Yeah. The question was, you see differences in the testing, but is that representing the natural situation, and why do you believe that is the case?
Very good question again. I think yes, it is representing, but of course I understand that different tissues have different expression levels, and therefore we have different transcripts. But when we do the mini gene assays, we already carefully look at which transcript we would like to approach, and what are, based on the in silico predictions, how we want to test them. And so, for example, with the SCA, we also used sushi cells, for instance, together with the hex cells. And so those cells are already known that they are quite representative of the biological processes. And so I think, yes, but not all.
True, I agree. And of course, you included the wild type.
Yes.
Which doesn't show the difference, the aberrant transcripts. Okay, continue on this. Of course, what we want to do with this knowledge is to see whether we can help the patient and reclassify the variants in this case. So you selected 4 splice variants in the SCA patients, of which in the end 2 showed a splice effect. And you choose 12, I think, variants in the cardiomyopathy patients, of which all show effects. But then in the end, the 2 that show effects in SCA, you reclassify. But in the cardiomyopathy patients, only 2 of the 12 you reclassify.
Yeah.
And you do kind of explain that, I think, but could you a bit explain to the audience here why you only reclassify 2 in the cardiomyopathy patients?
Yeah. So thank you again for that important question. I think, as I previously mentioned, that testing of splicing variations and therefore subsequently having the functional information is part of, but not conclusive of, the classification process. The classification process requires lots of other techniques that we need to tick the box for it to have a substantial evidence as together platform whereby we have
Hora, finita.
based on the ACMG guideline. In this particular case, we
I still have to ask you to give a short conclusion of your answer.
yes, well, in simple words, for the variants, we classified them as likely pathogenic or pathogenic. We had substantial evidence based on the ACMG guidelines. For those that we did not, they added value to the prioritization, but didn't add enough value to classify them.
Thank you.
Thank you very much. The doctorate board will withdraw for some time to consider its decision. The candidate. The doctorate board of this university, represented by us, after reading your PhD thesis and the accompanying propositions, and after hearing you defend it, has decided to award you the doctorate of the University of Groningen, which you desire. Your primary supervisor, Professor Simons, is authorized to award you this honor in the customary fashion.
I'm happy to accept this task assigned to me by the Rector Magnificus. However, before performing this task, I would like to address a few personal words to you. Dear Mohamed, very learned sir, many congratulations to you and your family on obtaining your well-deserved doctorate. Your journey from the start of your PhD studies to this special moment today has been an eventful one in many ways. As you so rightly pointed out in the last of your propositions, mankind has a cradle, which is in Africa. PhD studies have cradles too. And that one of your initial PhD project was created in 2014 with a grant application, which had been written by one of your co-promoters, Dr. Birgit Gadatz, and colleagues with the intent of looking at new types of testing using a new kit on the block called TLA that could possibly replace others in the genetic diagnostics of leukemia. And Zonnemeer, the grant agency, accepted our proposal, and this led us in 2015 to start searching for a suitable candidate for the position of PhD student on that project. You had finished your master in molecular biology and biotechnology at the University of Groningen, applied for the position, and your interview really stood out from the others in several ways, including your enthusiasm and your background and the way how you could convince us, and in fact your charming qualities, I have to be honest there. Well, you know, sometimes one is lucky and a particular project is a natural match with a particular PhD student. And in your case, it became apparent that another type of project might be a much better fit. So a PhD study is not only a way for a department to get a particular study carried out. Importantly, it also should take into consideration the learning goals of the particular PhD student. So we arranged, in fact, for the TLA project to be carried out by others, and we had a few long talks on what would be the best way forward. From the start, you had expressed your ambition to take particular scientific insight and hands-on experience with modern diagnostic genetic techniques back to Tanzania. And therefore, early 2017, we shifted focus to the diagnostics of germline variant studies, in particular diagnostic yield and different approaches of variant classification and follow-up testing. Indeed, this turned out to be an excellent move. You clearly felt more at home in that field. The new projects showed progress. You had made friends within the department and outside the department, of course. So in total, things were looking good. But then new developments arose, moving back to Tanzania and starting a new job there, for example. But these challenges to the project were overcome by the many hours that you put into your studies, the real dedication that you showed, and of course the continued support from many of your colleagues here in Groningen. And then the worldwide COVID pandemic struck. It struck hard at many people, including your own family and friends. And in addition to this huge personal impact for you and your dear ones in the world of science, it also meant closing down research labs and experiments coming to a full stop. So it was inevitable that this caused a delay in finishing your PhD. Delay is not always only a bad thing. It gave us time to produce much more mature chapters. And in fact, 4 out of 5 papers have been published so far. And the last one is about to be resubmitted after minor comments from the reviewers, so we will be fine there. Today, your PhD study has come to a successful end, a personal scientific milestone. Your family and friends, many of whom are now watching this ceremony online, must be very proud of you. Perhaps the thing we as your supervising team are most proud of when looking at you and your PhD is not just the content of the scientific papers. It's much more the way in which we have seen you grow in your ambition and initiatives to help develop genetics and healthcare in your country. And we are also so pleased and honored to have in our presence today Her Excellency Ambassador of Tanzania and the First Secretary. So Mohamed, you are co-founder and Secretary General of the Tanzania Human Genetics Organization. You are already involved in many genetics-related studies within your country, ranging from therapy for sickle cell anemia to pharmacogenomics and way beyond. Very recently, you have been awarded a fellowship by the University of California Global Health Institute with the aim of letting you train in the techniques needed for sickle cell gene therapy, if I understood correctly from you. As a department, we will be working with you, looking at opportunities for collaboration. So there will be no farewells today, definitely not, but rather I hope to talk to you soon. And in fact, I already saw that a next meeting has already been put in our calendar. So we will definitely talk soon about how to continue our collaboration. So Dr. Mohamed, again, our congratulations. We wish you all the best for your present and future career. And we are all looking forward to working with you in the years to come. Thank you very much. Please rise. By the virtue of the powers vested in us by law, in accordance with the decision of the College of Deans of the University, I, Rolf Simons, confer on you, Mohamed Zahir Alimohamed, the title of doctor and all the rights associated by law or custom with a PhD degree at the University of Groningen. Do you, as a doctor, promise to act in accordance with the Netherlands Code of Conduct for Scientific Practice?
I promise.
Thank you. Please take your seats again. Now that I have performed the task assigned to me, let me be the first to congratulate you on your PhD degree and to address you as doctor. Well deserved. Congratulations. The photo moment there.
As proof of this PhD, I will present you with this certificate signed by Director Magnificus, Secretary and Supervisors and sealed with the Grand Seal of the University. But before doing so, learned sir, it's my pleasant duty to congratulate you also on behalf of the doctorate board of this university on the degree you have been. We also present you with an album. It's a book about the city. And with this, I end this ceremony.